Opening shot, classroom setting voiceover, Professor, welcome back, students. Today, we will be delving into the fascinating world of biochemistry, specifically focusing on the complex topic of sulfur amino acid metabolism. This intricate system plays a crucial role in various physiological processes and has significant implications for human health. So, without further ado, let's explore the intricate web of biochemical pathways that govern sulfur amino acid metabolism. The methionine cycle is a series of interconnected reactions involved in the metabolism of the amino acid methionine. Methionine is an essential amino acid, meaning that it cannot be synthesized within the human body and must be obtained from dietary sources. Once ingested, methionine is converted to S-adenosylmethionine, SAM, which serves as the primary methyl donor in cellular methylation reactions. Methionine adenosyltransferase, MAT, is the enzyme responsible for the conversion of methionine to SAM. MAT activity is tightly regulated, as excessive SAM production can lead to increased cellular methylation and adverse biological effects. Various factors, such as feedback inhibition and post-translational modifications, play crucial roles in maintaining the balance of SAM production. SAM is commonly referred to as the universal methyl donor since it participates in a wide array of crucial biological processes. One of its primary functions is to provide methyl groups for DNA and histone methylation, thereby modulating gene expression and epigenetic regulation. Dysregulation of SAM-dependent methylation has been implicated in various diseases, including cancer and neurodegenerative disorders. Furthermore, SAM is involved in the synthesis of phosphatidylcholine, a major component of cell membranes, as well as the production of neurotransmitters and other biologically active molecules. As such, disruptions in SAM metabolism can have far-reaching consequences on cellular function. Inadequate availability of methyl donors, such as SAM, can lead to impaired DNA and histone methylation patterns, resulting in altered gene expression profiles. This phenomenon, known as epigenetic modifications, can have profound effects on cellular processes and has been implicated in the development of various diseases. For instance, global hypomethylation, characterized by a decrease in methylation across the entire genome, is frequently observed in cancer cells. This loss of methylation stability can lead to the activation of oncogenes and the silencing of tumor suppressor genes, contributing to tumor formation and progression. Homocysteine is a sulfur-containing amino acid that is generated as a byproduct of SAM-dependent methylation reactions. Elevated levels of homocysteine, known as hyperhomocysteinemia, have been associated with an increased risk of cardiovascular disease. The remethylation pathways play a crucial role in maintaining homocysteine at appropriate levels. Two main remethylation pathways, the methionine synthase pathway and the betaine homocysteine methyltransferase pathway, convert homocysteine back to methionine using different cofactors, such as vitamin B12 and betaine. Deficiencies in these pathways, often caused by genetic mutations or nutrient deficiencies, can result in homocysteine accumulation and subsequent cardiovascular complications. Hyperhomocysteinemia has emerged as an independent risk factor for cardiovascular disease. Elevated levels of homocysteine promote endothelial dysfunction, thrombosis, and inflammation, all of which contribute to the development and progression of atherosclerosis. In addition, homocysteine can directly damage blood vessel walls, leading to impaired vascular function. Multiple mechanisms have been proposed to explain the harmful effects of hyperhomocysteinemia on the cardiovascular system. These include oxidative stress, impairment of nitric oxide production, promotion of platelet aggregation, and alteration of the extracellular matrix. Strategies aimed at reducing homocysteine levels, such as dietary supplementation with B vitamins, have shown promising results in mitigating cardiovascular risk. Cysteine, another sulfur-containing amino acid, plays a vital role in protein structure, redox balance, and antioxidant defense. While cysteine can be derived from the diet, its de novo synthesis is equally important, as dietary intake may not always be sufficient to meet cellular demands. Cysteine biosynthesis involves multiple enzymatic steps, with cystathionine beta synthase, CBS, being a key regulatory enzyme. CBS catalyzes the condensation of homocysteine and serine to form cystathionine, which is further metabolized to cysteine. Mutations in the CBS gene can lead to CBS deficiency and homocystinuria, a rare inherited disorder characterized by elevated homocysteine levels and various clinical manifestations. CBS deficiency is an autosomal recessive disorder that impairs the function of the CBS enzyme. This deficiency results in the accumulation of homocysteine and its metabolites, causing significant clinical manifestations. Patients with CBS deficiency may present with intellectual disability, lens dislocation, skeletal abnormalities, and increased cardiovascular risk. Treatment options for CBS deficiency include dietary modifications, such as restricting methionine intake and supplementing with cysteine. Additionally, pharmacological therapies aimed at reducing homocysteine levels, such as vitamin B6 supplementation, may be employed. Oxidative stress, characterized by an imbalance between reactive oxygen species, ROS, production and antioxidant defenses, has been implicated in numerous pathological conditions. Glutathione, a tripeptide composed of glutamate, cysteine, and glycine, plays a central role in cellular antioxidant defense mechanisms. Glutathione acts as a reducing agent, donating electrons to neutralize ROS and protect cellular components from oxidative damage. Glutathione peroxidase and glutathione reductase are enzymes involved in maintaining the redox status of glutathione. Dysregulation of glutathione metabolism can lead to increased oxidative stress and contribute to the development of chronic diseases, such as cardiovascular disease, neurodegenerative disorders, and cancer. Chronic diseases, including diabetes, cardiovascular disease, and neurodegenerative disorders, are often associated with reduced glutathione levels. This depletion can impair the cellular antioxidant defense system, making cells more susceptible to oxidative damage and promoting disease progression. Aging is also accompanied by a decline in glutathione levels, leading to increased vulnerability to age-related diseases. Various factors, such as impaired enzymatic activity and decreased cysteine availability, contribute to age-associated glutathione depletion. Strategies aimed at replenishing or enhancing glutathione levels, such as dietary interventions and pharmacological agents, hold promise as potential therapeutic approaches. Taurine is a sulfur-containing amino acid that serves multiple physiological roles in the human body. It is involved in bile acid conjugation, modulation of calcium homeostasis, antioxidant defense, and regulation of neurotransmission. Taurine is essential for the development and function of the central nervous system, particularly during early life stages. It also plays a role in cardiovascular health, as it influences cardiac contractility, blood pressure regulation, and vascular function. Taurine deficiency has been linked to various health conditions, including cardiovascular disease, neurodevelopmental disorders, and metabolic syndrome. Sulfite oxidase is an enzyme critical for the metabolism of sulfur-containing compounds, specifically the detoxification of sulfite. Sulfite is produced during the breakdown of sulfur-containing amino acids, and its accumulation can have detrimental effects on the central nervous system. Individuals with sulfite oxidase deficiency, a rare genetic disorder, experience severe neurological complications due to the inability to eliminate sulfite efficiently. Symptoms may include seizures, intellectual disability, and neurological regression. Management of sulfite oxidase deficiency typically involves dietary restrictions and supportive care. Altered sulfur amino acid metabolism has been implicated in the pathogenesis and progression of cancer and metabolic syndrome. Aberrant methionine cycle activity, disrupted cysteine biosynthesis, and dysregulation of taurine metabolism have all been identified as potential contributors to these conditions. In cancer, altered sulfur amino acid metabolism can influence tumor growth, metastasis, and response to therapy. Metabolic syndrome, characterized by obesity, insulin resistance, and dyslipidemia, often exhibits perturbations in sulfur amino acid metabolism. Understanding the underlying mechanisms and developing targeted interventions may offer new avenues for prevention and treatment of these complex diseases. In conclusion, sulfur amino acid metabolism is a highly intricate system that encompasses numerous interconnected pathways and enzymatic reactions. Its regulation and dysregulation have profound effects on cellular function, gene expression, and overall health. By unraveling the complexities of these metabolic pathways, we gain insight into the etiology of various diseases and potential therapeutic strategies. Further research in this field holds immense promise for advancing our understanding of human health and developing innovative treatments. Voiceover, Professor, thank you for joining me on this journey through sulfur amino acid metabolism. I hope you found this exploration both enlightening and inspiring. Until next time, keep exploring the fascinating world of biochemistry.